So today we are going to discuss about gene expression. When we talk about gene expression, we already know genes are the hereditary materials present in the DNA. So genes, what they do is they basically produce proteins, and how it happens is DNA gets converted to RNA and RNA gets converted to proteins. So the process of conversion from DNA to RNA is called transcription, and the process of formation of proteins from RNA is called translation. So this is how your genetic information basically works. That means how DNA is able to produce proteins is through a series of steps, first involving conversion of DNA to RNA. This particular process is called transcription, and finally RNA produces proteins by a process called translation. This entire genetic process is called central dogma. So this is how in living organisms genetic information works. So today we were going to talk about gene expression. By gene expression, what it means is there are many thousands of genes present in an organism. So if all the genes are functioning continuously throughout its lifetime, that means there will be countless number of proteins produced, and not all proteins are required during the lifetime of an individual. Like certain proteins will be required during certain environmental conditions or the stages of an organism. So gene expression is basically controlled by the environmental factors. So depending on whether the factors are present or not, your gene expression can be studied under two headings, like inducible and repressible system. By inducible system, what it means is if you want to produce a certain protein, then what do? You add that protein in a medium. That protein acts as an inducer which acts on the genes to produce more protein. So in this case, the system is being induced to produce more proteins. Such an example is called inducible system. Now, if you want to repress the system, then what you do is a repressor produces a protein called repressor protein. This repressor protein acts on the genes, and as a result, your protein production stops. So, first thing we're going to discuss about inducible systems. So when we talk about inducible operon, we're going to discuss about E. coli. E. coli is a bacteria which contains many thousands of genes, and not all the genes are working at the same time. Only a certain genes are working at the required environmental condition. So in this case, inducible operon, what it means is a certain enzyme will be produced which is being induced to work to produce that particular protein which helps in the digestion. So galactose is a disaccharide made of glucose. Then what happens is galactose is digested by an enzyme galactosidase permease. This is an enzyme which digests the galactose. Now E. coli can grow in medium which contains galactose or does not contain galactose. So now how this operon works is there are three enzymes or genes These ZYA genes are responsible for the production of enzyme galactosidase permease. This P stands for promoter, I stands for regulator. So now your regulator gene produces a protein called repressor. Your ZYA gene produces the proteins beta galactosidase permease that helps in the digestion of galactose. Now what happens is, generally when the E. coli is growing in a normal medium, this repressor protein binds to the operator. As a result, your ZYA genes, they are not producing this particular enzyme. So that means the bacteria E. coli is not able to digest galactose. But now what happens is, if in the medium you add galactose, then what happens is, this repressor protein does not bind to the operator. So your ZYA gene produces galactosidase permease. That means now in this particular case, the E. coli is able to digest galactosidase. How this happens is, the repressor protein binds with galactose that has been added in the medium. As a result, this protein undergoes a change in its structure and it is no longer able to bind to the operator. So this first case represents the repressed state. That means your enzymes, that is your genes ZYA, is no longer able to produce the enzyme galactosidase permease. But in this case, the operon has been induced by galactose. Galactose combines with repressor, leading to a change in the conformation or the structure of the protein. As a result, your genes ZYA is able to produce galactosidase permease. So that means E. coli is now able to digest galactose. So in this particular case, galactose acts as the inducer. Now in this case, we're going to discuss about repressible operon. Now what happens is, if a particular gene produces a protein and this particular synthesis is not stopped, then the protein will keep on accumulating. So that means this protein, let's say, is an enzyme which leads to increased metabolism. So in order to stop the metabolism, this production of protein has to be stopped. In certain cases, what happens is, the end product of this particular protein itself stops the production of its protein. Such a system is called repressible operon. That means the end product of the final protein stops the production of its protein or its own self. So in this case, again, we're going to discuss about E. coli, a bacteria which can synthesize an amino acid tryptophan. This amino acid is needed for various metabolic reactions. Tryptophan is again produced by 5 genes. A, B, C, D, E. All these genes are responsible for the production of tryptophan. Your P is your promoter region, O is your operon, and your R is your regulator gene. Your regulator gene produces a protein called repressor. In the earlier case, the repressor was bound to the operon, but in this particular case, it is not bound to the operon. So what happens is, these genes A, B, C, D, E are continuously synthesizing tryptophan. So this tryptophan that is produced from the genes now binds with the repressor. That means tryptophan is now acting as a co-repressor. So your repressor and your co-repressor, the complex, let's say this is your repressor and your co-repressor is this one. So these two found a repressor co-repressor complex which binds to the operon region, and the tryptophan is no longer produced. So that means the end product of the tryptophan itself is stopping its own production. This is an example of repressible operon. The repressor, it alone cannot stop the system, but it complexes with the end product, that is your tryptophan, and hence stops the production of tryptophan. So this is an example of repressible operon. So in both the operons, either the end product was involved in stopping the process or in initiating the process. So your operons or gene regulation is controlled by the environmental factors. Like for example, in the first case, your lac operon, it was an inducible system. The inducer was galactose. As a result, your E. coli was able to digest or utilize galactose for various purposes. The other example, tryptophan belong to repressible system. That means tryptophan is an amino acid which is required for various metabolic functions. Tryptophan genes, there is A, B, C, D, E, were producing tryptophan. This tryptophan is a co-repressor that combined with repressor produced from the regulator gene. So your co-repressor co-repressor complex stopped the production of tryptophan. So this is an example of repressible system. In addition to this, you all know that DNA is a long segment. So if this is your DNA, not all parts carry genetic information. Only certain parts carry information. So if you divide the DNA, you will find that it consists of exons and introns. Exons are the ones which contain genetic information. That means this genetic information is utilized to produce RNA, and then finally proteins. But introns, they do not contain any genetic information. So within a segment of DNA, only exons are the ones which are utilized or necessary for information. Introns are not that necessary. So this is it. We will be continuing with Human Genome Project in the next class.